can officially start.
Okay, perfect. So welcome back to Biocentrica Presents. We've got Roy here for a follow-up conversation for understanding chemistry, manufacturing, and controls in drug development. So Roy, please take it away. For everyone else, hold your questions and post them in the chat, and we'll visit them during our Q&A session at the very end. Go ahead, Roy.
Hi, everybody. So I am Roy, just as what Penny just said, and I'll be talking mainly about CMC, but just some housekeeping audience guidelines. I know everyone's already muted, and of course, it's supposed to be designated as a professional environment, so please be respectful, courteous at all times. I have no disclosures. This is a informational session, so nothing will be proprietary information. So a little bit about myself and why I'm even doing this presentation, right? I just did one on clinical pharmacology last time, maybe 3-ish weeks ago. But I was really interested in CMC and I do have some experience in it. So to start out with, I have a bachelor's in general biology. This bachelor's, I did a lot of bench research. So you can think about like PCRs, Western blots, etc. And that's where I kind of got my foot in the door for more pharmacy related things. I then went to go get my pharmacy doctorate because my pharmacy doctorate is where I wanted to sort of step foot right into the industry world, do something different. There, I again was in a lab and I was in a variety of different things, right? Compounding lab, investigational drugs, and the key for this particular topic is drug product development intern. So that's sort of where my CMC career started. After that, I went to become a clinical pharmacology fellow, and after that, I was invited back to become a drug product. You can't hear me? Uh-oh.
I can hear you, Roy. Why don't you, if anyone can't hear, try rejoining the session.
Oh, okay. All right. So I'm going to keep continuing. But yes, drug product development scientist, and that was actually specifically within the CMC space. I was in that for quite a while. Went back into clinical pharmacology, and now I'm a clinical scientist. So to go a little bit more into what CMC is, CMC stands for Chemistry, Manufacturing, Controls. When I first started out, I didn't know anything about CMC whatsoever. And so a lot of my learning was actually on-the-job learning. CMC is mainly actually for everything that has to deal with the drug. So the drug substance, product development for the drug, any manufacturing concerns, quality is a very big issue, and regulatory compliance. There is a whole niche of it, essentially. You can go all the way from preclinical to commercialization for CMC, okay? And the goal for CMC is that you're ensuring this product safety, quality, and the big one is consistency as well. Now, there's quite a lot of practice areas, and I'm going to tell you now, this is going to be a very, very large and a very simplified, at least, version of CMC. So the drug development process mainly will start out in API. API will then move on to the drug product development side, which will be more formulation work. Then it goes into analytical development. So you can think assays, process chemistry and engineering. This would be a lot of, manufacturing from like small scale to large scale. And then regulatory CMC, of course, you need your regulatory folks. But because CMC is so large, usually regulatory affairs has their entirely separate CMC department, right? And the last thing is quality assurance. So of course, making sure the drug is consistent, it's safe for a patient, etc. So we'll start out on API. API is mainly the designing, synthesis, control of the API to ensure that purity, potency, and last but not least, the scalability. So when you're super early in this process, a lot of the times, if you don't know the drug is going to work, you don't want to make it in bulk, right? So you'll probably make a little bit of it, and then you'll end up scaling it up. But so in the very beginning stages, you'll be developing that synthetic route and any control strategies. So you can think back to organic chemistry when you are looking at a small molecule, for example, and creating a new molecule out of it. So those are all where those organic chemistry techniques sort of come in. for those people who are interested in organic chemistry. Then after that, this is, you'll do a lot more of the technical side. So particle size, impurities. Impurities can go anywhere among like, is there any metal impurities, any rubber impurities, anything at all, really. It has to be the most sterile process that you possibly can do. And that's very important for CMC. And what I was kind of mentioning a little bit about the scalability portion, it's that they need to be able to scale, obviously, to be able to supply to the rest of the world. And the impurity, optimizing that yield, obviously, to save money and increase the impurity or decrease, I guess, impurities of your specific drug product. So this is a little bit more of a jargon: critical process parameters and critical quality attributes, CPPs and CQAs. These ones are going to be more of how are you going to write, for example, an SOP from start to finish of making your specific batch. And then the quality is, as you can probably guess, it's the quality for making sure that the drug doesn't have any impurities, essentially. After that, there will be tech transfer. Tech transfer is what sort of technologies they use on the small scale, and being able to transfer that technology into helping manufacturing, for example, scale it up for the manufacturing part. And so that's sort of what the tech transfer will help with. Drug product development, usually after the very small scale, right? That API gives you a smaller batch. This is where drug product development will develop a formulation. What API gives you is a very basic buffer. They don't add much to it. Okay, so we're thinking about excipients, there's like microcrystalline, or if you're looking at large molecules, you have to think about pH. So a histidine buffer versus an acetate buffer, etc. Excipients wise, do you need any polysorbate? Or do you need any citrate? Stuff like that. And those all are done in DPD formulations. You will then select those excipients, you conduct these manufacturability studies. Fill finish is here, meaning fill finish is essentially you have your product and you put it inside maybe a vial, or you'll put it inside of, like, how would you put it into the vial? Like, those are going to be filling the vial and then finishing up that vial presentation, let's say. Like all the blending, the compression, those are more for small molecule tablets or capsules. The container closure system. Okay, so like what I was mentioning with the vial or even a bottle, you can look at a pre-filled syringe. These are all container closure systems. You need to decide what is going to be best in it. And then for DPD, I think one of the bigger studies that usually you'll do are these stability studies. Real-time and accelerated. Real-time is obviously just regularly at room temperature. You could also do stability studies in the refrigerator or in the freezer. The accelerated stability studies are usually at like 40 degrees Celsius because, as you all may have learned in previous biology classes or chemistry classes, when you heat something up, it'll degrade faster, right? And so that's how you will simulate this accelerated situation. So then your stability study doesn't need to go on for 2-3 years. And then the last thing would be these bridging studies for formulation or packaging changes. When I say a bridging study for formulation, that means if you, for example, have a large molecule and in the beginning you have an IV formulation. An IV formulation is actually very easy to do because you're just mixing it into like a bigger bag, let's say, of sodium chloride or glucose or dextrose, etc. But based off of feasibility, like if you want at-home administration, a lot of people go into subcutaneous, so under the skin formulations. This is very important because depending on which formulation you do, the excipients are all going to be different. But you need to bridge this discrepancy essentially and make sure, you'll prove to the health authorities that, hey, my drug product in an IV formulation works and is just as safe as if it was a subcutaneous injection. And that's what those bridging studies are mainly used for. Same thing with the packaging change. So for example, it was in a vial. Now you're going to put it in a pre-filled syringe for subcutaneous administration. The next thing is analytical development. Analytical development is going to be there throughout the entire process, okay? They are the ones who validate the methods. And these method validations are extremely important because this is how any health authority is going to say, yes, we agree your drug is stable based off of the specifications that you've given us and based off of the way that you've analyzed it. Like, we trust you based off of these methods, okay? And there's obviously these assessing what all of these things are, and those are all the same things that regulatory agencies also assess. And to validate these methods, you're really going to need to develop things. So HPLC, that's the high performance liquid chromatography. You can have gas chromatography. Many, many, many analytical methods are out there. And a lot of the times you're thinking like assays, these methods, a lot of them are actually proprietary information as well. So some of them are not publicly available. It depends really on your company. You can also have a patent for some of these methods, etc., which is actually very fun if you're very big on wanting to do a patent, for example. Characterizing the degradation. So the degradation of products, that kind of goes a little bit into the stability portion of DPD, right? So DPD would be giving the samples after they take the samples from a 3-month, 6-month study, and they hand it off to analytical development, and they analyze it to make sure nothing changed dramatically. And it's within specifications. And I'm not going to say specifications, I'm just going to say spec instead. And again, like I said, they will do the establishing specifications, release criteria to make sure, yes, this is going to be a safe product to release to the general public or even in a clinical trial. The analytical comparability for formulation or supplier changes. Sometimes, actually, you do not need to do a bridging study. And when that happens, it is usually a comparability between 2 batches. So when you're making a batch, okay, a really, really large manufacturing batch, you have to guarantee that these 2 batches are going to be the same, okay? Because the process that you've done might have been slightly tweaked, slightly different, okay? But it's not like the drug is different significantly. You just need to make sure that based off of batch 1, which was approved, batch 2 is the same. So that's where the comparability team comes in. The last thing would be providing any analytical support during those stability tests, the release, and the validation. The next one is process chemistry and engineering. These folks are a little bit more engineering oriented, right? They are the ones who do a lot of the fill finish. They'll do or help with it, and then as well as the tech transfers. They are essentially helping that scalability, because a lot of the scaling from small to large requires math. It requires actually a lot of engineering concepts as well. So you're trying to validate your manufacturing process, and you want to make sure it's reproducible, okay? These are the people who are going to create the SOPs. They're going to make sure that you follow these SOPs, and these SOPs can work from small to large scale. You perform these risk assessments. You can support pilot plants, GMP execution. So a lot of my previous process coworkers, they would go on site majority of the time if we were opening up a new site, or if we had a new fill finish or a new tech transfer. Like if it was the first time that the site did something, you essentially have to help execute. And the documentation, right? All the validations, you have to document, and mitigating these process risks. So for example, if your manufacturing plant, depending on the lighting that they could have, does your drug degrade? You need to think about a lot of this information when you're in process chemistry and engineering. The reaction time, how long do you keep it within a specific buffer? How long do you think this process would take from small to large scale? And then the tech transfer that I was mentioning earlier, making sure that it doesn't matter if it's from site to site. Like you could be all the way in Germany and you can be in the United States plant. They need to be the same, exact same. So these tech transfers need to pretty much piggyback off of each other and make sure it's the same. The next thing is regulatory CMC. This is actually pretty similar, honestly, to the CMC that a lot of PharmDs are familiar with, except they focus solely on CMC. These are going to be the ones who do the regulatory submission for drug approval, life cycle management. They specifically prepare this Module 3. Module 3 in an NDA, BLA, IND submission for United States at least, it's pure quality. Okay, that's the only thing that they're really going to be looking at. And they need to essentially submit this particular module to health authorities. They'll do a lot of these communicating with regulatory agencies, so the IRs, they look at regulatory guidelines, submission planning, and they are essentially coordinating everything with cross-functional teams and any data collection with CMC. Quality assurance is the last one, and quality assurance is the police in a way. They make sure that everything you did was correct and all of the GMP regulatory standards, everything was done according to any guidances that was published, right? And they do a lot of this oversight. So they do internal audits all the time and support regulatory inspections. They can definitely manage any of these like corrective preventative action plans, which is a CAPA, and change controls. They will be the ones to review batch records. So they do deal with a lot more paperwork. And as you can expect, it's because they have to ensure quality is up to par. And, they oversee a lot of the suppliers, GMP trainings, and then any documentation control. So CMC is, as you can tell, a very, very, very large umbrella. It's essentially CMC umbrella and then the clinical umbrella. But a lot of the times, there are so many different things within the clinical team or the CMC team that it's a little bit difficult to give a lot of a nuanced approach. So this, again, I just want to point out, is a very broad overview. So CMC responsibility-wise, they will ensure a lot of drug quality and consistency, like I mentioned. You'll be developing, controlling these manufacturing processes, and then support regulatory submissions, like I mentioned, your CMC RAs, designing, executing analytical testing strategies. They do have stability studies, so they have to still write the protocol just like a clinical trial would. They're obviously a little bit different, but they are protocols that they do have to adhere to. And then obviously your analytical team will be analyzing your samples. Managing GMP compliance and documentation, that's a lot of QA's role. And then leading these cross-functional technical transfers. So the tech transfers are a lot of like the process team as well. All right. So select skills necessary for CMC. This is not an inclusive list, but as you could probably picture, a lot of organic analytical chemistry fundamentals. This is so important. You need to understand how these studies are working. You need to understand what is happening to your drug. And the formulation, excipient compatibility, that's just a lot of reading papers, understanding, why would we add in sucrose, for example? It's a cryoprotectant. Manufacturing process design, scale up, analytical method development, process validation, tech transfers, and then stability testing and degradation pathways. So what possible pathways could this molecule degrade under? Can it degrade under light? Can it degrade under heat? Can it degrade under cold? Etc. A little bit more of like the regulatory quality. You do want to be familiar with any ICH guidelines because that is going to be probably one of the most important things that you need to know to ensure that you're running your studies correctly. And authoring, there is quite a bit of writing in Module 3 as well. It's a very, very large module. GMP principles, change control, lifecycle management is important because sometimes in the middle of a drug, like it's been marketed, etc., maybe one day you think, I want to go into pediatric patients, but pediatric patients are actually very small, right? And what if you have a device that pediatric patients can't use? It is up to CMC to create a new formulation, or it's up to CMC to create a new device, okay, for these pediatric patients. That is why CMC has to still be there for lifecycle management. any regulatory intelligence, global variations, and of course, you'll still have your IRs. So any questions from regulatory agencies, you'll need to know how to answer. Soft skills wise, it doesn't matter what functional area you go into, you need to understand cross-functional communication. So how do you explain this to your clinical colleagues of, hi, my stability batch, we can't allow a shelf life of 12 months. The question is why? And you need to be able to explain that in an easy to digest way to your clinical folks, because the clinical folks will not care about the why if it's too technical, right? They just care, why can't we have it for 12 months? And give me the reason as to why we can understand it easily. The writing documentation, like I mentioned with your 3, attention to detail. The attention to detail is honestly very important because everything that you do is going to be recorded and documented. Documentation for CMC will be important just based off of like a quality perspective and a safety perspective. Time management, yes, because there are going to be peaks and troughs of when you are busy. And sometimes everything is going to be a I needed this yesterday situation. Risk assessment and problem solving, and then adaptability, because sometimes priorities will change and sometimes the clinical team might actually have a different suggestion on your formulation. As you can probably tell, there are many, many challenges, but I will just highlight a few of them. So the first challenge is this manufacturing scalability and reproducibility. Because of the fact that you're going from small to large, let's just take a bowl of pho, okay, for an example. If you put piping hot soup, okay, in a bowl, obviously the cooling down portion might be different depending on the size of bowl that you have, right? More surface area means a possible faster cooling period, right? So that's essentially what scalability is. You need to make sure that once you put it into like a large vat, for example, it'll still all do the same exact thing that you wanted it to do. The reproducibility, for example, how many times do you shake the IV bag? It depends. How big is the IV bag, right? You could do a 100 milliliter IV bag versus a 1000 milliliter IV bag. But you might only be putting in like 5 milliliters of drug. And you need to make sure that it's shaken correctly, okay? The next thing is regulatory complexity across global markets. Because a lot of the times manufacturing sites are not just within the United States, not just within Europe, etc., there's different guidelines for each place. And there's going to be inspections from each regulatory agency. And that's why you need to work hand in hand with a lot of the regulatory guidances within the countries that you're globalizing in. And that is why your regulatory CMC folks are very important. Tight timelines and compressed development cycles. So this is very common actually, because clinical team will say, for example, hi, we've gotten, or even medical nurse, they'll say, hi, we've gotten some reports that my patient's been getting injection site reactions. We need you to make a new formulation. The thing is, to make a new formulation, you need stability studies. You need to formulate the new formulation, etc. And you need to do this really, really fast so then this drug can then be put back onto the market or put back with new clinical trial studies, etc., to help these injection site reactions. Maybe you change an excipient, right? And so this is what I mean by tight timelines. It usually comes up and you need to be able to plan very, very, very quickly. Supply chain and material variability is the last one that I have on here. So during COVID, which is a great example, a lot of people couldn't really get the supplies, right? Everything was expensive. What do you do when, your manufacturer went out of business due to COVID? Or what do you do when even the exchange rates, let's say, like from euros to USD changed? Now everything is going to become more expensive, and you need to figure out where to get the money from. You need to figure out where to get these products, where to get the materials to make your drug product from. And so that's a lot of operational running in the background. And I know a lot of people like the last but not least, education, career progression, and salary. I try to tailor this a little bit more to folks who are not PharmDs because I am a PharmD myself, but I understand CMC is not always PharmDs. So, education requirements, you can enter as a bachelor's, and that's the wonderful thing about CMC. It's very, very common to go into like QA for associate roles, manufacturing. You'll usually have a chemistry degree, chem eng, or pharmaceutical sciences. Very, very basic degrees. If you really wanted to go forward, master's degree is fine. The master's degree is when you kind of move a little bit more into the formulation role because you need to start understanding the chemistry, right? Analytical development and regulatory CMC. This one will give you more of that hands-on experience. And the last one is a PhD. The PhDs are by far the most people, okay, that I've personally interacted for. And you'll get more senior roles. You'll see a lot of them in formulation development. And these are going to be the thinkers, right? So why does my formulation work in this particular buffer? Why? You have to answer the whys when you've gotten to this PhD level, okay? And this is what I mean by you need to have this independent research and leadership skills for this. Okay, so for career progression and salary, this one is very, very variable, okay, I will say. Entry level, mid-level, these sometimes kind of overlap. And I specifically put the years of experience after your bachelor's degree, okay? The bachelor's degree, you'll see in a lot of job postings, is sort of like the baseline. And that's why these are going to be your associate, right? Then you're going to move up a little bit in scientist role as you get career progression. And a lot of the times for PhDs that I've seen, they always start out actually at the senior scientist role. Masters, you'll probably see them at this senior associate scientist or even analytical science. But if you have a PhD, you can skip quite a bit. But as you can see, the base salary range is in line with pretty much having a PharmD in the previous talks that we've actually had. So they do accordingly give you the salary range to be in line. As you get more into your years, you can definitely go into leadership, senior leadership. Compensation will increase accordingly. Higher insights. So what do we care about in CMC? We want prior experience. I think the prior experience, the best part about it, okay, is the fact that your lab experience is counted, essentially. Because if you've done wet lab, that's all very relevant to, right, analytical development. It's relevant to drug product formulation because you'll learn for how to pipette, how to run a PCR, this kind of information. And some other bonuses, I guess, is if you've done regulatory submissions, right? If you've done development, analytical development, method development, process validation, GMP environments, any scale-up activities. The next one is, of course, the scientific acumen. You just really need to understand chemistry very, very well. Like, you need to understand formulation. If you're able to do that, then it goes to show that you're probably very trainable because lab skills can be easily taught. Knowledge is a little bit more difficult. And you want to be able to interpret all of this data. So stability, specifications, and understanding what exactly is good manufacturing process, right? The GMP, process risk, and quality control. The last one is, again, soft skills. You really can't have a job without any soft skills. So you want to be able to write in a way that is clear, concise, and having that cross-functional, like I mentioned before, attention to detail, and being very flexible with problem solving. And being creative, honestly, is very important in CMC. So we are now wrapping up a little bit. Wrap up wise, you want to be able to build a little bit more of this foundational experience, right? So internships, so something like I did, right? I ended up getting a job offer back with them in October. after I graduated, and they were willing to wait for me for, what was it, 7, 8 months before I actually got hired on as a full-time employee at that particular company. So the internships are key because you could potentially get hired on again as a full-time easily. And co-ops are actually kind of special. Co-ops are like bachelor's, master's, and PhD students. I've never heard of it in a PharmD program, but essentially it's a 6-month internship. And you take you take a semester off of school, and you just work. Entry-level roles in QA, that is something that is getting more and more popular, I think, because it'll help you understand the data, because you need to understand how to analyze it, right? You need to understand that whatever the scientists said that they did, they actually did. And then hands-on experience, exposure to GMP documentation, stability testing, being able to understand this documentation, volunteering for cross-functional projects that have the CMC components. So even if you're in an industry position that isn't in CMC, it's likely that they will work with CMC in one form or another. So just see what you can do to collaborate a little. The next thing is learning the language of CMC. I will tell you now, there is a lot of acronyms in CMC. There is quite a bit to learn, and ICH guidelines will be your best friend. Understanding where CMC goes into, which will be module 3, and current trends. So current trends too in CMC right now, subcutaneous formulations are the go-to because it helps patients administer at home. They don't need to sit in a chair, like right in a doctor's office, waiting for their IV to finish. They don't need to be hospitalized. These are all very, very important for you to understand what a CMC trend is. So see what kind of drugs are being approved and see what their formulations are. The next thing is transferable skills. Scientific writing, like I mentioned, very key. Have you gotten a paper published? Have you been a first author, co-author, even an author in general? Very important. Data organization, attention to detail, and communicate how your background aligns with everything in a CMC job description. Those you can look online of what the job description, and then just sort of highlight your skills, finesse it a little bit. And if you're able to, being able to look right at module 3 sections, case studies, class projects even, you can demonstrate any understanding of CMC, which is possible. And with that, I have finished my talk, and I am open for questions.
Excellent, Roy. Thank you so much. Great presentation. We're going to start our QA session now. Thank you, Orphis, for creating a great list of questions as well. We're going to start off with a question from Farmer One. He asks, so there's a specific CMC QA, just like specifics. Wait. I'm not sure I understand the question. It says, so there's a specific CMC QA, just like specific CMC QA, or does QA usually deal with CMC? Farmer, can you clarify that one? Oh, like specific CMC reg. So like regulatory.
Yeah. So you will have specific QA for CMC because, right, and honestly, you'll mainly see QA more so in CMC than you do in clinical.
Okay, perfect. Our next question is, how often does CMC interface with other functional areas?
Every single day. It's necessary. They will interact with clinical all the time because if there's a clinical trial ongoing, you'll need to interact with them. And medical affairs is really important because, well, less so medical affairs. You will interact with some of them, but your main colleagues that you'll interact with are the clinical team because you'll be helping them out with any new formulation work, etc. Medical affairs or even clinical operations or any operations, it will be them interacting with, like, how do you get a drug shipped to a specific country, for example, or what are the manufacturing needs?
So on that note, you'd say there's a lot of knowledge necessary of other countries' sort of regulatory components in order to make that process smooth, right?
Can you repeat that?
Yeah, sure. So on the topic of getting things like drugs delivered and whatnot, their components, I suppose, for people in CMC, you'd likely have to have a strong knowledge of individual countries' regulatory requirements then, right?
Yeah, you do. Usually though, if it's already been approved, so for example, in like your module, a huge list of components, right, that you put into your drug, if it's been approved, you really shouldn't have too much extra regulatory hurdles in a sense. But in the beginning, yes, you might want to, talk to the regulatory agencies a little bit more because there are like specific USP standards for your drug product and for excipients.
Excellent, thank you. Timo asks a chemistry question: how do you mitigate charged species or any other impurity species in general?
So this will be like for charged species, that's kind of where your formulation comes in, right? You'll have a specific buffer. So let's say that, you want a citrate buffer, a little bit more acidic, or an acetate buffer, histidine buffer. These will all have different charges in your buffer based off of your pH, right? And so those ones are going to be how you really deal or mitigate any of these charged species wise. As for impurities, a lot of the time it's just filtration, very basic. You can do ion exchange resins to remove any of these impurities during your purification process as well. And yeah, a lot of like the pH again that I mentioned. But other than that, process optimization, you can do synthetic steps, right? Designing these steps to avoid impurities. And that's where your like chemistry background and knowledge is important.
Excellent, excellent. Orphis asks, what did you enjoy most between immunology, oncology, and endocrinology? How much retraining did you have to go through for each different area?
I think endocrinology was probably the hardest for me to learn because I had to learn that one from ground up. Oncology, immunology, they're actually not too bad because if they're a large molecule, because I usually worked in large molecule, and those ones are very straightforward. I feel like oncology is probably still one of my favorites just because of how simple it is. But you will need to retrain. Like you will need to understand clinical if you do move into a clinical role. But if you're only interested in CMC, you actually don't need to know too much about any of them, like any of the clinical components at least.
Excellent. So you can rely on your cross-functional partners to fill in the gaps, so to speak.
Yeah.
Excellent. All right, we've got another question here. Since tech transfers need to be the same across plants, no matter where they are in the world, how often do they encounter regulatory differences by country, and how are those challenges resolved?
So a lot of the times you'll probably have like different batches or lots, and they're available in different regions. And so that's kind of where, right, your comparability results or your comparability team will need to do a lot of the heavy lifting. You have some of them will require like region specific comparators, and the tech transfers are very important in that sense as well. As for how they're resolved, it's kind of the same thing, right, with the bridging studies. So you have your reference drug, and you'll do a bridging study to ensure that, yes, this is all going to be the same if there's any issues. And you can have like a United States, EU test product, for example, and maybe just a PKPD analytical study. You can also, on another hand, is you can actually choose a globally sourced reference and which is accepted, right, across multiple markets. So, for example, an excipient, it can be used based off of the guidelines of each country. You can just buy this one excipient that you know will be accepted in these particular countries. That's usually how people are able to mitigate any of these issues between plants and countries and so you'll ship them right from a very specific manufacturer. Just making sure that you have your specs all the same as well. So right, that analytical development part, testing, ensuring that it's going to be the same, all very important.
Okay, and is that process similar with biosimilars as well?
Yeah, it is. Biosimilars are a little bit special. You'll have like a reference drug, and because biosimilars though are technically they're a new molecule in a sense, you need to go through all of the hoops to make sure that it is the same safety and efficacy profile. It's not as easy to have a biosimilar reference product versus a small molecule one. And so those ones will go through different hoops, I think, than maybe what that specific question might have asked.
Gotcha. So similar to biologics for all intents and
Yeah.
purposes. Okay.
Yep.
Timo asks, what tests can you conduct that allow you to simulate the shelf life of a product?
So this is going to mainly be in DPD, right? They're the ones who undergo all the stability testings. You can do the accelerated and stress testing. Those ones are mainly going to predict long-term stability. So you'll have elevated temperature, elevated humidity. If you think that there's actually photostability issues, you can put them under very, very strong light and then see how it degrades there. And you will usually use a model for like degradation rate. You'll assess shelf life, support any label claims, and use equations as well to estimate the shelf life that will be under normal circumstances. The thing is, even if you do an accelerated study and you have a tentative shelf life, you do need a concurrent or like in parallel have a normal shelf life, like a normal condition shelf life to prove, hi, my model, my equation, they were all correct. Okay. The long-term stability testing is mainly the gold standard. You will more than likely have to ship it off-site in order to prove that, in order to have them like use contractors, etc., to have them pull the sample, because those studies can go on for 3, 4, 5 years even, and that'll support final expiration dating. Intermediate stability testing can really help support clinical trials because it's not as long-term. And freeze-thaw cycles. So for example, you have a large molecule product. Depending, for example, the COVID vaccines, we all heard about them being in like -70 fridges, -80 fridges or something. And when you take it out, you have to thaw it. But the thing is that stresses a molecule really badly. And so you'll do these freeze-thaw cycles to ensure that nothing is happening on a more molecular level to your drug. And then there's something called an in-use stability testing as well. That one is very common because you will be testing the stability of your drug in the particular container that you want it in. Okay, so let's say that you have an IV product. The product is now going to need to be, the product is originally in a vial. The vial product has to go into an IV bag, right? You need to test what that in-use stability is. How long can it be in the IV bag before it starts to degrade, essentially, and in a simple way. As for time points, let's see, you could probably do all the way up to 6 months, like I said, all the way up to 5. It's very normal to do a 6-month study just for like clinical trial purposes.
So honestly, what you're saying is that DPD is the most fun part of CMC.
Okay, I can only say that because I have the most experience in DPD, so I'm very familiar with DPD studies, with all that. Um, I'm not very familiar, right, with the other ones that I mentioned. I know of them because I had to personally work with them, but those ones I think are a little bit more technical. You won't find as many in there. You'll find more like bachelor's, master's, etc.
Gotcha. I'll say this from a construction background, breaking things down and trying to test the limits of materials has always been my favorite part of the role. So I feel like I would have translated very well to DPD based on how you describe it.
Yeah.
All right, we have a question from Orphis. Do you have any advice for someone interested in getting into API development or DPD? Orphis states they're interested in going to school for toxicology. Where's the best fit for that skill set?
So if you want to go into toxicology, I think the clin farm route would be a little bit more useful for you because in toxicology that they're focusing a little bit more on, I guess, the drug itself and like the mechanism behind the drug. CMC in my eyes is more on how do I create this drug to be stable? How do I manufacture this drug? So it's a lot more chemistry related, whereas toxicology is more biology related. So if you like API development would be more chemistry based. So that I think it really depends on what your interests are then at that point. Like based off of the information that I've explained to you, do you feel like toxicology would be very beneficial? Do you feel like maybe pursuing more of a chemistry degree would be more beneficial for you? But again, right, it really, really depends on what you want to do and why you want to do it.
Thank you so much. Farmer One writes, "Hi, Roy. I was wondering, do you have any experience interacting with colleagues or teams focused on CMC policy advocacy?" Farmer One states they've met someone in CMC policy during an internship, and they're really interested in how companies engage in shaping regulatory expectations at that level.
I have personally never met anybody who was focused on CMC policy advocacy because they don't interact that much with the scientists. It's more of like a leadership decision, so those are completely separate meetings usually. I don't really know how companies really engage in it. I'd assume lobbying, but I'm again not very familiar.
Okay, we can move on then. Timo writes, can you go into why biosimilars may be so different if they don't get the interchangeable indication, even if they use the same process as the branded medication?
So the biggest difference with biosimilars really is going to be how are these biologics made? It is dependent on your living cells. Okay, so you'll probably hear some words thrown around like a CHO line cell, something like that. These are made in living systems. Okay, like these proteins are made, and so even the smallest mutation in a cell line, even the media that you use, the temperature, the pH, purification methods, this can change like a whole bunch of the chemistry. So chemistry being maybe glycosylation, how the protein even folds, change charges on the drug. All of these could actually be very, very specific towards that particular strain that you're using, right? And technically, manufacturers don't know the process used by the brand name drug because they're all proprietary information. Like this doesn't usually ever published anywhere. I would be very surprised if it is published because it's supposed to be as like a well-kept secret. I know that Regeneron has a process I think it's Regeneron. They have a process where, for example, they've patented, right? And now it's very difficult to use their particular process, okay? And you can technically still sell it, right? You could sell your process to somebody, but it's very difficult for, just like a random drug company to think of it off the fly. And of course, there's going to be these minor differences. There can be differences, but overall, you just want it to be similar enough with like the structure, your function, any efficacy, safety, immunogenicity is a really big logic. So
Makes you wonder how they discovered the miraculous nature of the Chinese hamster ovaries, I suppose.
yeah, they are very interesting cells.
Let's see. Can you recommend any literature for someone interested in CMC topics, let's say beyond the ICH guidelines?
The way that I learned was honestly just reading a lot of papers. There's actually manufacturing papers out there of what are some CMC considerations for AD. There's a lot of papers that people publish, and there are working groups actually where they do kind of like a joint guidance that they publish. So the big companies, they essentially hop on a call together and write this guidance document. And those I think are going to be very valuable.
Say again, Roy. I think you cut out at the very end there.
Oh, those are going to be very valuable. Like those working groups where a lot of different pharmaceutical companies or scientists will sit in there to sort of have like a best practice guideline.
Gotcha. Okay, so kind of similar to how regular guidelines are developed.
Yep.
Excellent. Orphis asks, do you, Roy, hold any patents?
I do not, unfortunately. I know a few of my old coworkers do.
Remind me again, because I'm a little unfamiliar with it, but when people tend to hold patents within companies, they receive some sort of dividend of the sales, correct?
I don't think so.
Maybe in
At least
drug discovery?
I think in drug discovery, possibly, but when you get a little bit later on, it might not be as common. But also, I don't think I've ever asked any of my coworkers, like my previous coworkers, about that. So it may be.
I'll have to follow up with some people and figure it out because that would be very interesting to know.
Yeah.
Right. Orphis asks, how often does the crystalline properties of an API impact the reproducibility or scaling of manufacturing?
It could impact it, but there also should be some mitigation strategies um, that are put in place. So that's kind of where your process development team comes in. And let me think about some examples because I'm not as familiar with manufacturing. So as for these, they can exist in multiple forms, right? So that's kind of how like a crystal in general, and you'll have different of these, like how soluble these different crystalline forms are within a different pH range. Okay. And if it does end up changing unexpectedly, unintentionally, you can have unintended consequences. So, can this drug end up being administered safely? A lot of the times, you'll actually get a lot of these crystalline crystals within like a drug product IV wise, but it really should not happen that often because if it happens, you will likely not get regulatory approval because this ends up becoming a safety issue, right? There are specs for how much you can have, but from what I recall, they should be very, 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 very low, non-existent.
All right, sounds like a very detailed answer. Thank you so much. Hopefully that answered the question. We're going to go into the next one. How difficult do you think it is for a non-clinical person in CMC to hop to another function?
I don't think it's that difficult, actually, at least based off of what I know from my other coworkers. I've had people from Discovery come to DPD, DPD people go into Discovery. We've had people go into toxicology. So it's not impossible. It just depends on what your end goals are, I think. So just as long as you have the experience, you have the knowledge, you have the drive, and you're able to accurately explain why you're moving, it shouldn't be impossible or very difficult.
Have you ever seen anyone jump to med or commercial from CMC?
Mm. I think these ones are a little bit unfair though because they were jumped. I've never really seen a PhD jump as a lateral. I've seen them take a decrease in their title for sure.
Right, excellent. I don't see any more questions. Unless anyone can type really quickly. Roy, I want to thank you again for your time. This was really engaging. I learned quite a lot. CMC has always been a bit of a mystery to me. And I think you did a great job. And to the audience, thank you so much for all the engaging questions. And Roy, thank you again.
Yeah, anytime. All right. Have a good night.
Good night, everyone.